Cool. So now let's start. So let's say we're going to get a bunch of attack-seek data, right? Are we good to? Cool. So what does attack-seek data tell us? Like what questions can we ask? Remember? Open chromatin? Yeah. So one of the things about attack-seek is that it gives you a read of open chromatin, genome-wide, which in a way is very powerful, but at the same time, on its own, it doesn't really mean anything, right? Likely there's a transcription factor bound nearby, or there's a regulatory event, but what event that is, what gene it's near, is all sort of a challenge to annotate, to deconvolute that data, and try to sort out what those regulatory events might be. But before getting to that stage, we have to sort of process the data. So we get back paired end sequencing, right? We're going to pick up sort of at that annotation point, but sort of maybe someone can, so we get all these sequencing reads back, right? Like FASTQ files, then what do we do? Like from there? We have, right, even before that, like QC, yeah, align. Yeah, you could do QC now or N probably after, but yeah, align. So you can use sort of any DNA aligner. I've been using BWA. So it's a typical paired end analysis. You sort of take the FASTQ files for read 1 and read 2, and then align each separately, and then you do a step to bring them together. And you end up with reads that represent areas of open chromatin. Right? And so often there'll be sort of something, but can I erase it? So we're going to have, a pair. Hard to see. There'll be sort of insert in the middle, right? It's like RNA-seq, and where the two pairs between the two pairs. So that in total will represent sort of our fragment length, as we call it. And one of the first things you can do is to look at the distribution of that fragment length, which was the chart that Olivier showed. And if anyone's interested actually in the code to do that, it's pretty simple. I'm happy to email it to you. But it gives you this sort of distribution of reads corresponding to where distances of nucleosomes apart from one another, right? So when you have nucleosomes within between nucleosomes, you get a bunch of reads, and then where you have a nucleosome, which is like 147 base pairs, you won't have reads because the DNA is protected there. And then again, between two nucleosomes, you'll have reads. Essentially, you have a nucleosome in between, and then the paired ends coming between them. So that's sort of why you see this distribution that's like phase-like. It's called like mononucleosomal and dinucleosomal and trinucleosomal reads and so on. So what's after aligning? Someone mentioned it before. Now it's like all our reads are aligned. We get BAM files or whatever. Yeah, peaks. So in a lot of ways, attack-seek is similar to ChIP-seq data. Kind of make a few hacks in a way to max 2 to count peaks. But essentially, we're getting the same output, which are regions where there are distinguishable increases in read count. So to sort of pick up, now we're going to kind of pick up from where you'd be after you've gone that far, you've gotten your peak calls out of max 2. So there's a useful package called rtrack layer. So we're going to, if we haven't already, call library, and you should have called library for actually all the packages. So then we want to bring that file in. So if everyone wants to, could probably execute down through these second lines. If there's, so basically we're bringing in the output from max2, which is called a bed file. And there's some additional information in there, and we're sort of telling R what sort of additional columns we have and how to treat that data. This. How's that? Should be good? Cool. So then I think it's worth, let me do this myself too.
I got an error, could not find function import.bed.
So that's probably make sure you run library and in parentheses rtrack layer. And if that gives an error, you need to be called BIOC light. So what do we expect to see in a file with peaks? Like what sort of information do we expect? Yeah. Yeah, there'll be like, right, there'll be the indicated chromosome, there'll be areas that are peaks, right? How big is a peak going to be? Like, yeah, it sort of depends, because it's paired in sequencing and based on that fragment length distribution, the entire regions end up being about 500 base pairs. That's like the median, but they can be longer.
Does it call them narrowers?
So that's sort of a, people sometimes refer to like ChIP-seq transcription factor peaks as narrow peaks, and like histone marks, which are like larger regions, as broad peaks. It's a little bit confusing in that ATAC-seq can kind of do both for you in some ways. So, but it's probably more of like the narrow peak. The data looks probably more like a narrow peak. But in any case, that's sort of where that comes from, and that the name of the file. So bed files have a number of varieties and flavors, and this flavor is called a narrow peak file. But it's standard enough that you can load it up into IGV, and it'll load that, and it'll bring in the scores and stuff too. So, right. So here's our file that we brought in, right? Giving the regions for each peak, and then it should give us information about each peak, and what we're most interested in is the negative log of the p-value. So it's, a p-value will be, right, the smaller the p-value is better. In a way that's a little bit more intuitive, we could take the negative log of that number, and then that'll give us sort of a linear range of higher numbers being more significant. So it's sort of a common way to deal with, sort of turn a p-value into a score. So what do we do next? Now we've brought in, we have our peaks. What can we say so far about? Well, first of all, I should say that, so these are peaks called from, the data is from a lymphoma cell line, and it's from the chromosome 18. So that's basically the input. So what can we do with the data now? Like what are there any questions we can ask as far from where we are? Or? Yeah, right. That's probably probably the best way to start. I would say so. So there's a sort of a number of ways we can do that, but we're going to need genome information, right? So based on the coordinates, where's our nearest gene? But thinking about that, like, what are the sort of implications of doing that? Like, let's say every region of open chromatin, we say, oh, that's the gene it's associated with.
You could have multiple genes associated with different regions, or the same region.
Yeah, right. Or right, for one gene can have lots of peaks nearby. What about like where we might find peaks? Like where would if there's regulatory events happening, where would those be in reference to the gene? Like,
Promoters and enhancers.
yeah. So where would we usually find a promoter? Right, which is sort of like the transcription start site, so the beginning of the gene. It's usually called 2.5 kb. Is it just in back, or is it either direction? I always forget that. But anyway, it's right in front of the gene. So there we'll be pretty sure that that area of open chromatin is probably associated with something to do with that gene. Enhancers, we tend to make the same assumption that for an enhancer, so for every peak, we're going to assign it a gene, and we're going to do that by distance. Right. Yeah. So that's the difficulty in making that assumption, that it may not be. Sometimes, right, it may not be that that's the gene regulating it, and there's some more sophisticated approaches to try to predict regions that are farther away than promoters, what those regions might be regulating. By the way, if we were to do that, like, do you think we could do that just with a TAC-seq? How would you? Yeah? We'll see how we can kind of integrate other data types to sort of try to get more information about our data. So the next lines of code, basically TXDB is like a database type in R that holds information about, it's like a transcript database. It allows you to pull out annotation information, which you can then connect to sort of the broader annotation databases. So everyone up to this point? Any questions? Yeah.
The negative log of the Q value.
Mm-hmm.
I guess goes to the calculation. What kind of ranges are you considering?
Yeah. So Olivia mentioned it a little bit before. So, but I've been using the p-value and then actually using IDR, which is in a sense it's similar to what Q-value tries to get at, but with multiple samples, you kind of rank where the p-values fall to set your threshold for where you want to set a cutoff for what you call a peak or not. I usually call peaks very leniently at first, so I can get a lot of. So I'm sure to sort of capture all the data at first. So I usually set a p-value of 0.01 or 0.001, like so, 1 to the negative 3. And that'll sort of give you a lot of noise too. The other way to do it is to set a threshold more like 10 to the negative 7. It'll give you sort of more or higher confidence peaks. But in the sort of IDR analysis, you want to kind of include some noise so you can get a sense of where your data becomes less reliable. Cool. So there's a really nice package called chip peak Anna, and it's easy to sort of run these things, but what are some of the considerations we might want to make when we assign a peak to a gene? So the idea being that for each peak we want to assign it some gene symbol. So how do we do that? But we talked about like the nearest, right? Nearest distance to the nearest transcription start site. And then another question is where we need to find a point, right, somewhere on the peak. So a good strategy to do that is just to take the middle of the peak. It's likely your region of near the highest read density. For other considerations, you might want to take the one end of the peak, depending on sort of what your question is. But you can sort of set all those parameters here, and then we use our annotation data that we brought down from the database, and then as a way to sort of interpret it, it's easy to remember gene symbols, and it's not so easy to remember like entree IDs, right? So we'll give each entree ID. So genes typically, yeah.
Is the nearest location always a valid way of assigning peaks to TSS?
I don't know. What do you think? I mean, when might it be, or when might it not be? I think no. It's probably not.
Do you know roughly how often it's an incorrect way of doing things?
Yeah. So sort of we were talking about before. At promoters, it's probably pretty reliable. We can look at what some of the ataxi data looks like later, but if you see a big region of open chromatin that's 10 base pairs away from the start of a gene, most likely it's the regulatory machinery that's regulating transcription there. But enhancers, which seem to be seeming more and more like the more important regions to focus on. There's a recent paper that looked at that assigned enhancers based on RNA-seq expression, and then they sort of said how often it wasn't the nearest gene. I'm thinking, I think at least like 70% of the time it was correct around for that cell type, but that's still not a great if you're missing 30. So the other thing is that I think often you can have peaks that are in an intron that are regulating a nearby gene also. So that's sort of another caveat. So one question we can ask are what genes are assigned that are in a promoter region, are pretty reasonable to say that it associated with that gene. So we'll pull out. I don't know like how interested you guys are in like the details of the R code. If you are or just want to run it, like either way is fine. So so for this code, for instance, let's hit or select ANO like this, and run that and see what you get. And of course, you have to run the commands to make it work. So it's going to give us a whole bunch of information. So R has a data structure called the genomic range. It's very similar to what a bed file might contain, although maybe more powerful in some ways. And so all of our sort of groups of regions in R that the way I've been working on it are in this data class called a genomic range. And that's sort of for each each peak that's going to give us, let's see, this specific region, and then a bunch of information related to the gene it's near, how near it is, the score of the peak. Could give us what other types of information might you want to know about a peak? Yeah, like how wide, how tall, right, in a way, like how many reads are within the whole area. What else? So you want to, you have a region, a promoter, you want to take a stab at what transcription factors might be binding there. How might we get a sense of that, or ask that question? Sorry? Yeah. Yeah, that's a great approach. It's not, I didn't include it in this trope, but again, I'll make sure you guys have my emails if you want to look at sort of similar analysis. There's a program called Homer, and so you can, that will return a similar result as this, like a genomic range with individual matches for motifs that correspond to binding sites, right, of these factors. What other ways could we get like a sense of maybe like existing data? It's sort of another, there's sort of an enormous amount of data from ENCODE and other experiments, ChIP-seq experiments that are available that we can look at overlaps of peaks with our called peaks and get a guess. So that's some of, all that sort of information you can store in genomic ranges. And so you end up having it's a nice way to sort of put everything into one holder, basically. So let's look at this next line of code. So we're going to pull out what are those gene symbols nearest. So this is going to tell us on chromosome 18. And what does that show? Are you guys able to pull up the gene symbols? Any sort of players that look familiar? I think maybe BCL2 is in here. Anyways, as it turns out in ATAC-seq, for the most part, most promoters you'll get a peak. At least it's sort of fairly typically expressed genes. Yeah, so quantifying is another great sort of way to go to ask more questions. So we can count reads, sort of similar to RNA-seq, and compare. So that'll give us sort of a number of read counts that are within a peak. There are all sorts of caveats to that and sort of ways to deal with that data. I think there's a little bit of that towards the end of the tutorial, but. So we pull out the peaks that are nearest to promoters. What about other peaks? And like, what would be our guess for how many peaks are like just at promoters? How many? Where else would we find typically find peaks. Enhancers, right? Maybe in introns. So it might be interesting to look at what the breakdown is in our data. How many of the peaks are at promoters? How many are at enhancers? Enhancer is sort of a specific term. It sort of implies that it's probably functional, I think. But you can at least say like there's a peak that's, you know, further away than 2.5 kb from a gene. So let's go down to 61. Or like that. So the next thing we're going to plot what that distribution looks like. What the percentage of how the peaks come out. And what do you think?
There's where we think everything's going to come out.
Yeah, how do you think, or yeah, when you see the plot, what are you surprised by what you see, or is that? Actually, there's a pie chart that we can look at too before. Is there anyone the code for this the pie chart? Does that come up? So it's a line that starts with pi. So after we've annotated, right?
So what
Yeah.
does overlap mean? Does it overlap with each?
Yeah. So let's see. What are you looking at specifically? Oh yeah. So this is sort of looking at for a peak, where is it related to a gene? So if it's within, you know, it's between the TSS and the end of the gene. If it's like inside, right? Upstream would be promoter or anything else. That's sort of directly from the calling. You can sort of annotate this type of information further with genome information. So that's sort of the next analysis step here, and we'll. May take a few minutes to run. That should be. Is everyone able to generate this bar plot? Yeah. So there's a lot in like introns and enhancers, right? It's like a lot of the peaks. And this is also, I think, just on chromosome 18. And for whatever reason, I think the distribution is even stronger against promoters on this. So a regulatory heavy chromosome, I guess. But
So this is only checking in genes, right? It doesn't count like intergenic regions.
Yep. Yeah. So you might have to expand it a bit. This far plot is introns, right? Or this is intron, sorry. Intergenic is, so there are areas that are further away than 2.5 kb, right, between genes. So you could call that putative enhancers, I suppose.
Yes.
You sort of want a little more evidence that there's a regulatory event happening there to call it an enhancer. But likely. You know, not many exons like we'd expect. Is everyone up through that point? Okay. I think the code should work, but you never know. So what other questions could we ask? Like with respect to available data sets, what could that tell us? What kind of, say you're interested in just characterizing this cell line, these LY1 cells. What kind of ChIP-seq experiments might we look for? Anyone? Antigen receptor? I don't know. Yeah? Yeah. Yeah, so histones are probably would correlate well, right? Like there should be some histones that are related to regulatory regions, some that are related to enhancers, some related to promoters. And then transcription factors is probably a little more specific to the cell type we're interested in, and even more like what sort of questions we want to ask. But if you're lucky, so for this cell line, there's data available for this cell line for a number of these experiments, which is nice because across different cell lines and tissues and things could look very different. I think people are seeming to see with where enhancers are and whatever. So I included bed files, and the script will pull it up automatically. But anyone heard of gene expression omnibus? Yeah, so they hold a lot of information. So basically, I downloaded the one paper that used this cell line and looked at a bunch of different ChIP-seq experiments and histone marks and various things. So basically downloaded that and took their areas that they called peaks and asked the question, where's the ATAC-seq data capture those regions? And hopefully, one idea is that are regions that sort of are likely to be enhancers. So that particular mark is like a HK327 acetylation. So it might be a pretty good readout of where there's regulatory events happening. So if there's an area that's pretty likely to be an enhancer and also has open chromatin, like that might be somewhere where we could do more specific motif analysis, for instance. What factors are there? How does that location change? If we treat ourselves with inhibitor or between a related cell type. So the next line of code is basically this next bunch of code. You should be able to just run it all if you want, or we can go through any of the code. What are you guys interested in? Like details about the code, or more like what does the result look like? And
I don't know what the code is.
okay, cool. So let's see. Yeah. So first, basically, I create an empty list called ChIP-seq database, ChIP-seq DB. A GRanges list is like a data structure. It's basically a list for genomic ranges objects. Remember our peak calls file was a genomic range. So a genomic ranges object. So a ChIP-seq peak calls are also going to be a genomic range. And they're all going to be related because they're all on, they're all relative to the same genome, hopefully. Well, and if they're not, you'll have trouble. And sometimes they're not actually. I think this data was HG18 when I downloaded it. So it's actually it's simple, but you have to sort of translate that over to HG19. It's called liftover. But in any case, everything's in one place, and so that sort of allows us to compare overlapping regions and sort of annotate things according to each other. Sort of puts everything in one container with hopefully some variables in common that we can relate, right? So going through, we're going to say like we're going to do a loop in R, and it's not loops aren't always the best thing, but for this, it's a relatively small number of things we're looping through, and within it, we're doing more. So I had made this file called tracks, which gives us like the file name for each of these bed files of peak calls, and like a name for it, BCL6 or HK34, whatever. And we're basically just going to loop through, and then as before, using our track layer, we're going to read in that peak calls, the bed file, and then the associated name of what it is, the factor. And then for each, this is sort of the key line, the sort of concept of the code is we create this empty list, a gerances list, and then for each genomic range instance, we sort of put that in its own container. So we sort of assign, we end up with this GR, that's the genomic range of a given experiment, and then we sort of put it in one slot in the ChIP-seq DB list. And we sort of go through all our experiments and slot them in, basically. Then, if the code works, we'll get something like this. It'll say G ranges list, and then you can see what that list contains by calling names and the name of the list. So we'll say names, ChIP-seq DB. You get that, and those are all the peak files that we've brought in. Sometimes I think there's some R, so a few replicates of things. I basically just brought it in as it was annotated in the paper. Any of these look like familiar players? Things might be interested in regard to, the questions we can ask. I mean, that's always another thing. Is like, what question you want to ask about the data, right? But these are probably some pretty important regulators for B cells, BCL6, and then these histone marks. Anyone have a sense of what these histone marks are? What do they mean? Yeah. So I've just learned all this stuff recently. But H3K4 methylation, I think, so that's typically at promoters, and it's not found in enhancers. And then one that's H3K27 acetylation, that's a mark of enhancer regions. So is P300. So we'd be particularly interested in, if we want to use our TAC-seq data to get a sense of where those open regions are. And then, in addition to sort of having a global outlook, what's like one sort of, maybe the most important, the greatest thing about TAC-seq, right? What makes it actually probably the most powerful as data? So related to what samples. So at TAC-seq, you only need 500 or 5,000 cells at sort of a minimum. Usually it's more like 50,000, but that's super low. For DNA-seq, you need 50 million, and that's really right out of the incubator with the best lab technique on their floor. The hope is that a TAC-seq will enable us to look at these types of readouts in tumors and in tissues, and in a way that's a way that we can do genome sequencing on clinical samples now. And if we learn more about regulatory regions, it could be part of the sort of tool set we have for precision medicine and for understanding drug targets or experiments on a more wide basis. So sort of think in a way, you have this TAC-seq data. What could you say if you didn't have, if you had some of this information? What kind of predictions could you make based on, now we have this cell line that has all these great annotations. What about in a real B cell? Like sort of how could we apply the regions we learn about? So those are some of the questions I've been struggling with, for better or not. So let's sort of finish up this. Any sort of thoughts, questions about this sort of stuff?
So I guess
Yeah.
if the list were very long, I guess the loop
Uh-huh.
will probably take too long. So is there an alternative code?
Yeah, so you could, I didn't, but you could very easily, well, somewhat easily, because there's a lot of variables going on. Like the apply functions, so basically R executes things as vectors, but although that's not always the case with apply functions, but it does execute things in parallel if you use so there are ways, but I think it's like 5 seconds or something for this particular code, so it's not I could wait that long, but yeah, sometimes running loops in R, if it's like something big, you'll end up like having to come back in a month. Like it's. So sort of. Let's sort of maybe go down and. There's a lot of code in here just to make basically a data frame structure of this. So that's a typical data structure in R that you can plug in to plot and to sort of relate variables in a way that you'd typically analyze them. Are variables categorical and all this sort of data science-y type stuff. So we're going through all that. Let's. I'm happy to talk more about it or answer questions, but I feel like let's maybe execute down through the plot and if it works. Oh, it's because it should work on yours. Is anyone able to pull up this plot? Like, what do you see? Yeah, right. The resolution's kind of squished on mine, and it's like falling error. Yeah, that's true. But let's see. I can do is, everyone in? Yeah, no, not. Is it calling errors, or are you seeing a plot?
So what's on the y-axis?
So let's actually look more at the code exactly. So in this code that starts with overlap table, OL table, everyone see these lines? So we're basically, the important function here is we're calling find overlaps. So that's good to give us regions in one, like the first file, that those regions that overlap, that have an overlap in the second genomic range. So basically we're going through each ChIP-seq experiment and saying for each peak, is this peak overlapped by an ataxic peak? Is it contained with, is it overlapped? So, and then within that you can say like what percentage is the overlap, or how many base pairs do you want the overlap to be? You can even take overlaps that are not actually touching and say, give me an overlap if two peaks are within 500 base pairs.
So we're just looking to see how well these other data sets from other transcription factors overlap with the ATAC?
Yeah, yeah. And the idea being, if we're, some we'd expect, right? So what might we expect to be captured well? If we're really getting a good readout of open chromatin, yeah, so right, so some of those histone marks. So I wish I could pull this up, but let's see. Wait a second, I'm going to pull up the. That's going to be really small. Okay, hopefully I can pull it up now. It's a bit mungled, but. Everyone got into this. So yeah, this is giving us the overlaps, and so, in a way this is getting to this point in analysis. I sort of had looked at, well, what's the reverse, right? If a taxi was just, if we were using whole genome sequencing, probably we'd probably see more in some of these, but we'd probably see really high overlap too, right? So this is giving you like sensitivity, but not really specificity. But assuming that our taxi calls are fairly specific, which you probably could see if you looked more in the data, and you could sort of formally address too. The overlaps, seem pretty strong. Any thoughts? We sort of had the advantage of in these overlaps, they're all from the same cell line, and actually all from the same. This paper was from Olivier and Ari Melnick's lab, so it's also from the same lab. But primarily the cell line probably helps. So I think, let's say we did it on a different cell line. What markers do you think we would see less overlap, or where might we see differences? Yeah, the transcription factors, totally. Yeah, BCL6 is definitely not active as a regulator in all kinds of cells at all times. It's not in B cells either, right? These are actually specific lymphomas to have dysregulation in BCL6 and related pathways. It just as a guess, I would maybe think some of the enhancer regions might be more specific to cell type, and then some of the promoter regions, maybe more overlap between related cell types, so less differences than enhancers. But that's just a guess, really.
Is it normal that K27 and K9 have so different overlap with the attack?
Yeah, I think so. I mean, it is to some extent the first time some of these experiments have been done. Although I'm sure what I think people have probably looked at is DNA-seq overlap with HK327, HK39. So that would be a cool thing to look at. Did they see similar overlaps? In some cases, I think some of it probably depends on how the marks were called, right? And like what was the stringency on those calls, and if you took like the most highest confidence H3 K27 peaks, for instance, I would hope that the overlap would increase, but these attack-seq peaks too are fairly stringent. So we're taking peaks that we can see at least 2 biological replicates in a given cell type. So you could start out with like probably 120,000 or so called peaks, and you'll see some reads there. But that number ends up coming down to 50 to 60,000 that are sort of the most distinct features. So I thought maybe we'd take lunch now, but and then there's some more stuff we could do after. You could also pull up. There's the BAM file for this. You could pull up in IGV if you have it. If you don't have IGV, you can download it, or I can help you with all this stuff. Lunch, but that will give you. In a way, I probably could have done that first, but Olivier mentioned and how important it is to sort of see the data, right? Like, what do these overlaps look like? Which I think is probably, it'll probably give you a sense of what these overlaps mean. But you could pull up all those bed files too in the ChIP-seq database folder and sort of inspect what they look like. I didn't include the coverage tracks for the ChIP-seq, although you could probably download something similar in IGV. But if you load the BAM file, it will give you a coverage track. Any questions? Fascinated, confused, hungry? Cool, thanks. I'm around. Yeah.